I'm going to hand over. I'm going to hand over to Kelsey in a minute to do a more formal introduction of Janice, and then she will chair the meeting at the end with questions. But just to remind everyone to please, if you are requiring SACNAS points for this webinar, to please put your name in the chat and indicate that you need that. If you could put your full name, that'll help us to get you that certificate. So thanks everyone, and over to you, Kelsey.
Thanks, Glynis. And thanks very much, Janice, for giving this webinar for SAAB. I first met Janice when I was a postdoc at Syracuse University in New York, and Janice actually gave a job talk there that was just amazingly awesome. And she was subsequently hired into the department, but has since moved to Queen's University. And Janice did her PhD at the University of Toronto with Spencer Barrett on mating system evolution, and then did a postdoc at Duke University with John Willis in North Carolina. And so her research interests are focused around understanding the maintenance of phenotypic evolution and adaptation, mostly in the context of plant reproduction and life history strategies. And Janice has done some really awesome work in the last couple of years. And so it's been a lot of fun to kind of follow her papers and how her lab group is going. And I'm really excited to have her here with us today. So thanks, Janice.
Thank you. So thank you, Kelsey, for that introduction and for inviting me to give a talk in this webinar series. It's especially near and dear to my heart because I am South African originally. So I immigrated to Canada as a teenager. But it was really my formative years spent in the Fynbos and in the Karoo and in the Drakensberg that instilled in me a passion for nature, plants, and variation. So it's especially special to be back giving a seminar to you back in my office in Canada. So as an evolutionary biologist, I'm fascinated by the diversity around us. And it's amazing to me that evolution has produced such a spectacular variety of solutions to a single core problem. And what I mean by this is that at its essence, life is about going from one generation to the next. And so I find it incredible that there is so much diversity around reproduction, around this process. And I think this is nicely summarized in this quote here by Harper. If the plant is only the means by which a seed produces more seeds, the variation in efficiency of the process are extraordinary. And everyone here probably works on plants, but of course, this could be generalized to all organisms. It's not a problem specific to plants. And so it's this efficiency and variation in the process of going from one generation to the next that really drives a lot of the research in our lab. So my talk today is going to focus on the diversity and evolution of life history strategies in particular. We also do a lot of work in my lab on pollination and mating system variation, but in the interest of creating sort of a cohesive talk, today I'm only focusing on the life history work. And like life history traits, things like survival, age at reproduction, trade-offs, allocation strategies, are really evolutionary solutions to ecological problems. So the ecological problems are things like limited resources and risks of mortality. Life history theory has a rich history of mathematical and formal conceptual development, but most of this work has been centered around explaining phenotypic evolution and not really focusing on the genetic mechanisms underlying that. However, life histories evolve as a result of the interplay between extrinsic and intrinsic facts. And so my work and the work in our lab really seeks to integrate these approaches to ask if we can identify the evolutionary processes and their genetic mechanisms underlying the life history variation. And in particular, we're always focused on natural variation and the variation that segregates both within and between populations. And what are the mechanisms that cause that variation? So we use this framework of life history variation to address fundamental evolutionary questions. And so within this talk, I will discuss processes like selection for divergence versus the effects of gene flow, which will homogenize populations. We'll also talk about the evolution of quantitative traits and some of the underlying mechanisms and architecture. So things like how many genetic loci contribute to the trait variation we're seeing. How do those genes interact? What are the genetic correlations and covariation among traits that can potentially constrain evolution? So it's these types of fundamental evolutionary questions that we're at the core interested in, and then using all of this life history variation to try and address them. So I use the species Mimulus guttatus to understand the evolution of life history strategies. And Mimulus makes a great study system for these purposes because it comprises two ecological races or ecotypes. So there's annual and perennial ecotypes. And these two ecotypes co-occur throughout most of the native range in Western North America. So unlike many systems of ecotypic variation, this isn't about a clear latitudinal gradient. It's not like in the south of the range, they tend to be annual, and in the north, perennial. They really are co-occurring throughout. So what I show in this map, the green points here show known localities of Mimulus guttatus to give you a sense of its range. And then the red and blue points show collections that we have where we know that the populations are either annual or perennial. And so you can really see this, that the two ecotypes coexist throughout the range. And so instead of sorting out by some large scale gradient, the ecotypes sort out at a much smaller scale based on water availability. So the perennials, which I show in the left here, occur in environments that stay wet year round. So rivers, lake margins, coastal regions, where the continual water facilitates a vegetative persistent throughout summer and into autumn. In contrast, the annuals occur in environments that dry out seasonally. So these could be just upslope of the perennials, but they're in seeps that are fed by snowmelt, for example. And so because of seasonal drought that will set in late spring or summer, there's a premium on flowering early and setting seed before the environment degrades. So we know that it's this difference in water availability that underlies these annual and perennial strategies, and yet they can co-occur at a really small spatial scale. They're completely intercrossable and compatible. There is no genetic isolation between these two different ecotypes. And yet, as you can see in this photograph, and I'll show you others later, they really do look quite different in both their environment and also when we bring them into a common garden. Okay, so we're going to start thinking big about large-scale genetic variation between the annual and perennial ecotypes. And then I'll focus in more to think about the maintenance of phenotypic variation within and between populations. So range-wide, and then at small scale, what's going on with variation. So the core questions that I'm going to address in my talk today fall under these topics. First one being, what are the evolutionary relations between the annual and perennial races of Mimulus guttatus? What maintains their phenotypic and genetic differences? And how does this inform our understanding of the maintenance of complex polymorphisms within species? Okay, so I'm going to jump right in here to the first question, asking how are the different life history ecotypes related? And this work is a bit older now, but it nicely sets the stage for the rest of the talk. So I want to kind of come back and tell you about this project. So this work was done in collaboration with Alex Twyford, who was a postdoc in my lab and is now a lecturer at the University of Edinburgh. So we collected leaf tissue from 69 populations range-wide, and I show the specific locations of these in a map of Western North America, where the annual populations are shown in red and the perennial populations are shown in blue. And we used Illumina sequencing with reduced representation and very stringent filtering to sequence these 69 populations. And then we applied this strong filtering so that we could use both phylogenetic, traditional phylogenetic approaches, but also population genetic approaches to understand the variation amongst these populations. And so you can, in terms of the phylogenetic relations, you can imagine that there's two potential scenarios. The first one, we might find relationships like something that I show on the left here, where we have a single group of perennials and a single monophyletic clade of annuals, and they kind of separate out nicely in these two monophyletic groups. Or you can imagine a scenario where there's sort of the two ecotypes are interdigitated within a phylogeny, and maybe there's some other pattern in terms of how these populations or groups are related. So this is really the question we wanted to address, is which of these two potential phylogenetic relations do we uncover when we look at these populations? So just to be clear, we're using a phylogenetic approach, but these aren't different species, right? This is at the population level. Okay, so keep these two potential trees in your mind, and I'm going to jump right into the results for what we find. So which of these two patterns did we discover? Well, we very clearly found no phylogenetic relations with life history. So we do not find a single monophyletic annual and perennial clade. Really, they're interdigitated throughout the range. There does seem to be this one group here that is largely all perennial that I'll get back to in a minute, but otherwise they're really kind of going back and forth in this phylogeny. So what pattern do we see here? So if we take this exact same phylogeny, and here I've got the branches color-coded by red and blue for annual and perennial, but let's instead color code them by geography. So same relationships, I'm just changing the colors here. If we color code it by geography, we actually find this really clear pattern of geographic structure. So what we're finding is this clade at the bottom, that happens to be at the bottom of the tree here, which I've color coded in green, that represents the southern part of the range. And then we have this clade of orange here, which on the map you can see represent these coastal populations that really just run north and south up the California, Oregon, and Washington coastline. And then we have a break here, and this second clade up here represents a divergent group that spread north following post-glacial melt. So all of North America, all of Canada and part of North America was covered in ice during the last glacial maxima and started receding about 20,000 years ago, and it took up until about 12,000 years for that ice and cold front to recede. And the red line on the map shows the extent of ice coverage. But in fact, the cold front from that glacial maxima spread down to the Siskiyou-Klamath Mountains, which is really where we see this break between the green and purple groups. And so we think that for most of its evolutionary history, the species was restricted to the southern part of North America, and then it only spread north following the movement of the cold front north and then the glacial spread. And you can see in these purple and blue clades, this really clear stepwise pattern north as populations migrated north. I also, on this figure, at the very tips of the phylogenies, depending on the size of your screen, whether you can see it, are an open or closed circle. So those are the annuals or perennials. And we found that populations in general were most closely related to the population that was geographically nearest to them. And so we find this pattern of adaptive divergence between annuals and perennials despite gene flow. So there's a lot of gene flow between populations that are geographically nearby, which is why we have this geographic pattern. So there's all this gene flow and admixture going on, which I show with this tree mix figure, which specifically shows admixture between groups. And yet when we grow these plants in a common garden, like the photographs on this slide show, they maintain these really different types of where the annuals really have very little vegetative growth, and the perennials are kind of larger and have a lot of leaves and stolons. We also find this clear pattern of isolation by distance that's just sort of wide of and if we switch instead and the genetic architecture of this, what we've done here is we've scanned across the 14 chromosomes of Mimulus guttatus to identify the regions of particular variation. So our y-axis here is FCT, which is a variation of FST, which looks at differentiation between groups. And then we're looking at comparing all the annuals from the perennials and how different are those ecotypes. And so points that are around the 0 line is very, so for example, very little difference are more differentiation between the two groups. And you can see that in this particular region on linkage group 8 here, and actually this region of linkage group 5 as well, we have exceptional allelic differences. So 40% of all of these outliers localized to this region on chromosome 8, even though it only makes up 3.5% of the SNPs that went into the database. So something is going on this region on chromosome 8 that underlies differences between the annuals and perennials. And in fact, we weren't particularly surprised by this because we know that in this region is a chromosomal inversion. So a region of the chromosome has switched direction that was described by David Lowery and John Willis in 2010. And so we know that the annuals tend to have one orientation of this inversion and the perennials have the other. And so we wanted to look at differences within this inversion versus the rest of the genome. And so when we quantified genetic variation across the genome wide, so this is another way of thinking about genetic variation, where each vertical bar here represents an individual, and it's a clustering algorithm that groups individuals that have genetic similarity. And so when we color code these by life history, again, we see no particular similarity between annuals and perennials. So on the left here are all the annual populations, but they don't tend to have more annual genes, for example. But we again see the strong signal of geography when we group these by geography. So that's genome-wide, again telling us that across the genome, there's lots of gene flow and admixture between these groups. But now if we focus in on just that inverted region of chromosome 8, then we discover that within that inversion, annuals tend to be really similar to each other, and perennials tend to be really similar to each other. So again, this inversion seems to be a region that's particularly different between all of our annuals and all of our perennials, and not with geography. So now the geography pattern falls away in this particular region. Okay, so we've sort of identified at this large spatial that annuals and perennials genome-wide have a lot of gene flow between them. This inversion, we know, is a particular region where the annuals and perennials are different. And so some of the genes underlying these clear differences between the ecotypes probably fall in that region. What I want to turn our attention to now is thinking more about those particular traits. So what are these phenotypic traits that differ within and between the annuals and perennials? So for this part of the talk, we're really going to focus in on the key differences in the life cycle between annuals and perennials and how they perceive seasonal changes differently. So we want to, if you think about annuals, they have a short life cycle that rapidly transitions from seed to a juvenile phase. Well, that part might not be rapid, but once they're in a juvenile phase, they rapidly transition to flowering when they receive the correct cues. Perennials, in contrast, have a much longer life cycle, and they typically have to cycle within and between different environments. So they receive and perceive winter sequential times and need to reset from flowering back to vegetative growth. And so we were really interested in these differences in the stages of growth and how selection at one stage could influence subsequent stages, and how these different ecotypes time these transitions between growth and reproduction. So what we wanted to begin by addressing are the implications of overwintering as either a seed or a rosette. So again, remember, the annuals are really only transitioning once, while the perennials are cycling repeatedly. And so it's this sort of overwintering stage that we wanted to understand. This work was done in collaboration with a former postdoc in my lab, Matthew Rubin, who's now at the Danforth Institute. So what we did, I'll just remove that slide, that bit for a bit, and tell you what we did. So what we did here is we used about 80 populations of annuals and perennials from across that range. So much of the same populations that I've already showed you. And we grew them in growth chambers under different, where the seed either experienced cold or it didn't. So we wanted to kind of replicate winter with these seeds. And then we grew them up and quantified whether or not the individuals flowered. And so what we found, again, annuals are shown in red and perennials in blue. We found that the annuals flowered, so our y-axis here is the proportion of plants that flowered, and they flowered regardless of whether they experienced cold. And this makes sense because the annuals have a single opportunity at reproduction. Even if the environment is suboptimal, they should just transition to flower and make the most of it. Well, we found with the perennials that if they received zero stratification, so they did not experience any cold at the seed stage, there was a significant reduction in flowering. So they stayed vegetative, which suggests that they're not going to flower until they perceive the correct environmental cue, because it's better to keep growing vegetatively until the season is appropriate. So that was for seed. And then we were also interested in looking at the same effect on the rosette, so the vegetative plant. What's the requirement for cold to break dormancy and induce flowering for these groups? And somewhat similarly, we found that the annuals flowered regardless. Again, even if they're in the wrong environment, they just flowered because it was their one shot. While the perennials had significant reduction in flowering if they did not receive vernalization or cold at the rosette stage. So this really clear difference between the groups. So that we've done quite a bit of work on this vernalization. I'm not going to show you the results of that, but we ended up quantifying the genes that underlie this very clear pattern and found that it was due to genes in the SVP, short vegetative phase, which are genes in the vernalization pathway that have also been characterized in Arabidopsis and other plants. So we were able to get down to some of the genetic mechanism for this particular trait. What I want to focus here on in this next bit is some of the variation in germination within a single population. So as well as these differences between annuals and perennials, we also discovered that within a single annual population, there was extraordinary variation in the size of seedlings at the start of the season. So the photographs I show here were taken at a field site on Vancouver Island, British Columbia on April 4th, which is our spring season. And you can see in the top panel that these relatively large rosettes, which have clearly overwintered, and so they've germinated in the preceding autumn, versus these tiny little seedlings that had just germinated. And so within this annual population, you've got these two cohorts, and we wanted to understand what effects this had within the single population on flowering. So this was the work of an exceptional honors thesis student in my lab, Taylor Middleton, who has moved on to go to medical school. And so what Taylor and Matthew did was set up a growth chamber to replicate a full environmental season at this location on Vancouver Island. So we downloaded data from Environment Canada for every hour of the day for a full year, and we programmed the growth chamber to mimic these conditions in real time. So we had humidity, daylight, and temperature within this chamber to fully mimic the setting. And then we watered the plants according to what we knew from data loggers to be the soil moisture at the site. So we did our very best to replicate field conditions in a growth chamber. Of course, there's caveats to that, but it allowed us to isolate the seasonal effects without all the noisy stuff of real nature, like herbivory and other disturbances. So what Taylor and Matthew did was to germinate seedlings at the fall and at the winter, at the spring, to replicate these different cohorts and then track their growth. And so what I'm showing you here are these different groups. So in orange here shows you the start of the experiment where we had fall germination. So this was our mocked September timing. And then the blue cohort here are the following spring germination. So we accelerated winter a little bit, or a lot. Need the plants to sit under snow for 4 months of the year. But otherwise we went with a single day as a single day. So we got these two cohorts of germination, exactly like the photo I just showed you. And then we looked at the timing of flowering. And what you can see is that, perhaps not surprisingly, the fall cohort flowered earlier and had a longer duration of flowering. And then the spring cohort flowered much more rapidly, but they did overlap. So the spring cohort caught up to some extent and overlapped flowering. And then death was highly synchronous. So again, this was an annual population where death is really about drought setting in. And so regardless of how big these plants in, once drought set in, they all senesced very synchronously. And so we wanted to kind of understand this difference in flowering time. And so what I show you here are the means from families. So we had multiple individuals from genetic families so that we could parse out these differences in plasticity versus genetic variation. So the single points here, for example, this orange point and the blue point that the line connects to were full siblings. So they came from the same genetic family. And so what you can see is that the fall plants have a much longer, a much broader distribution of the start of flowering time, which you also saw on the previous slide. The spring plants flower much more synchronously. And because of this high synchronicity in flowering, technically there's no genetic variation in the spring cohort. So selection actually wouldn't be able to act on flowering time for those plants. What effect does this have on fitness? So in our growth chamber, our measure of fitness was flower number, and we actually found very little difference in flower number. Both fall and spring cohorts had extraordinary variation. So individuals differed in flower number, but on average, they were really similar. So despite these different growing season lengths, those fall plants have a much longer growing season than the spring ones. Total flower number is the same. So how do those spring plants achieve this? How do they catch up and make the same number of flowers? Well, we think that they have different growth strategies. So I could switch slides here quickly, but I want you to see that I've completely changed the data here. So now I'm showing you leaf number in the first panel and leaf length in the second panel. And what you can see is that the fall plants make more leaves. So they're bigger plants with more leaves than those spring plants, but actually those leaves are smaller. So the spring plants have larger leaves. And so what we think that what's happening is that the spring plants germinate later, have to rapidly transition to flowering. And so instead of investing in new leaves, they're using the fewer leaves they have and just making them bigger. And that may allow them to photosynthesize to an almost equivalent rate and have the same resources to invest in flower. So these different growth strategies within a single population. Okay, so the last bit of my talk here, I want to transition to thinking a bit more about flowering time, but also some other aspects to allocation. So I've started to show you in that previous slide the effects of leaf number and leaf size and how this may affect flowering. But one of the biggest striking differences between the annuals and perennials is not only leaf number and leaf size, as you can see in this photograph here with the annual on the right, which makes almost no leaves and tiny little ones, versus this perennial on the left. But also perennials make these stolons, these above ground runners that facilitate clonal growth. And that's what really allows these perennials to persist for multiple generations. So they spread clonally in the environment. And you can see in this photograph on the bottom of 4 different perennial populations that there's a lot of variation in stolons, even within the perennials. So what we tried to identify in a whole lot of our research, and I'm showing you some of it here, are whether there are trade-offs between flowering and clonal growth. So this is sort of testing key fundamental ideas in plant evolutionary ecology and life history theory about having a fixed pool of resources, and you can either have one strategy or the other. So in this case, you can either flower early and fast, or you can grow bigger vegetatively and clonally. And so we wanted to explore whether those trade-offs played out within populations and between these ecotypes. So on the left here, I show you data from 274 individuals from 78 populations. Again, annuals in red and perennials in blue. So this is now across the range, again, pretty much those same populations. What's their variation in flowering and clonal growth? And on the x-axis, I have the number of stolons, or those clonal runners, and then on the y, the days to flower. And you can see that the perennials in blue have a lot of variation, but in general, make more stolons and flower later in the season. The annuals, in contrast, make 0 or 1 or 2 stolons and tend to flower much more rapidly. On the right, I show you some work that formed part of the master's thesis of a student, Kelly Schmid, who is currently a postdoc at Cornell University. And for this part of her master's work, she looked at the same genetic correlations within a single perennial population. And we were really surprised to uncover the same variation within a population. So again, it looks like there's all of this segregating variation, both within and between populations. And I will, in full disclosure here, draw your attention that the axes have changed. So we're not quite recapitulating the entire range of variation, but even within the single population, where these points represent different genetic families, we're uncovering the same trade-off between stolons and dazed flower. So we really wanted to kind of hone in on this, these two traits and understand them in more detail. And so one of the approaches that I used was to use a classic crossing and QTL, quantitative trait loci approach, to try and understand the genetic differences. So I took an individual from one of these typical perennial populations and an individual from a typical annual population, and I crossed them to generate segregating F2 recombined progeny. So I crossed them to make an F1 and then self-pollinated that F1 to make a large number of segregating F2s. So this is sort of a very traditional crossing design. But then what we did is we sequenced the genome of the two parents and all of the offspring. And we could then scan across the genome to use the sort of quick and dirty approach to map the loci underlying the differences in these traits. So you could imagine that for all the loci that differ between our annual and perennial parent, I've just color-coded these in red and blue across the genome. And then if we sequence our extreme early flowering pool and our late flowering pool, we would expect that all the regions like over here that have nothing to do with flowering are just about a plant being a plant. We wouldn't necessarily expect any differences. But then when we get into regions that underlie differences in flowering, we would expect our early flowering pool to have more of the annual parent genes, our late flowering pool to have more of the perennial parent gene. So this tells us then that the genes for flowering time are in this particular region of the genome. So we used that kind of approach for this QTL mapping. We had 2,000 progeny, so these recombined progeny that we phenotyped and then genotyped. And what we found, again, what I'm showing you here are the 14 chromosomes or linkage groups of Mimulus guttatus. And then the squiggly lines are much like, I'm just going to skip back a slide here if I can. Oh, no. Oh, yeah, the squiggly lines are much like these squiggly lines shown here, scanning across the genome. And so all the regions here where it hovers around the 0 point, there's no significant QTL or no significant quantitative genes contributing to these quantitative traits. But in regions where they're really different, like up here or down here on the bottom of linkage group 14, that suggests that our two pools are have different genes underlying those traits. And so we did this separately for the pool of plants that flowered early and late, and I show those QTL maps in orange, and then the pool of progeny that had lots of stolons versus very few stolons, and I show that in green. And what you can see here is that the two traits, the flowering and stolons, have a lot of shared genetic architecture. These messy squiggly lines, I admit that they're messy, but they tend to be really similar. Like where the flowering ones suggest genes underlying flowering, so do the stolons. And so this suggests that the genes controlling flowering time and stolon production might be shared. This may sound strange if you're thinking about how could, a vegetative runner have the same genetic architecture as flowering early. What we think is going on is that these are really genes underlying meristem fate. So does the meristem take on a reproductive role or a vegetative role? And so it's not so much about flowering versus stolons per se, but about the type of growth that the plant experiences. And so that may underlie this trade-off between these seemingly disparate traits. Okay, so that's what we find in growth chambers. Many of you, and certainly myself, are much more interested in natural variation and what's going on in nature, which is where selection's really happening and where fitness actually matters. And so what we really wanted to do was take all of this back out into the field and ask about fitness and allocation in the field. And so with Matthew Rubin, we devised a very large, possibly overly ambitious field experiment where we created recombinant crosses between those different genetic groups that I showed you in the first part of the talk. So we're sort of putting everything together here. So we selected a field site on Vancouver Island, which I'm showing with my arrow here on the map. I know there's a lot going on this map. And we had a perennial parent from Vancouver Island that we crossed to an annual parent from Vancouver Island. So those were both local to the site, but differed in ecotypes. But then we also crossed that same perennial parent to 2 other perennials from the different genetic groups in the south. So we're recombining both the genetic groups and the ecotype, the annual perennial groups. We kept all of those 3 cross lines separate. We, in the greenhouse, did 2 generations of crossing to reshuffle all the genes for these 3 different genetic groups. And then we put 300 of these recombined F4s back into the field from each set. We also had 20 replicates of the parents, field replicates of the parents, and F1s. So in total, we had about 1,800 plants in this field site that we then monitored for 2 years of growth, flowering, overwinter survival, stolons, and seed set. So we measured a whole series of both sexual and clonal fitness. I show you all the traits here that we did for year 1 and year 2. Because Matthew is a total glutton for punishment, we also actually measured flower number, fruit number, seed number, and counted seeds. Mimulus guttatus seeds are tiny. They're about the size of a poppy seed. So this is an incredible amount of work. So we are still parsing our way through all of this data. The phenotypic data is done, but what we also did is collected tissue from all these plants to sequence the genes to the Q genes under trait and fitness in the field to see if they match up to what we were getting in the greenhouse and whether these trade-offs play out in the field. So I'm going to show you all the phenotypic data. In part because of the pandemic and because Matthew and I both moved to different positions, we have not made as much progress on getting the genetic data analyzed and complete. So unfortunately, I can't show you that, but I'll tell you a bit about it at the end. So here are the phenotypic data for our different groups. So I've really kind of broken this down. All those 2,000 plants are kind of broken down into 4 groups here. So this is our northern perennial parent, which is local, our coastal perennial, our inland perennial. So the first 3 here are all the perennials. And then on the far right here in red again is our local annual plant. So these are the parents, and we measured clonal growth in the field. And you can see, not surprisingly, the perennials had more clonal growth than the annuals, okay? What I'm now adding on to this figure are the F1s between the crosses between these groups. And in all cases, we found sort of the F1s had like hybrid vigor. So they had more clonal growth than either of their parents. So they were elevated for clonal growth. But also that the F1s all showed the dominant maternal phenotype. So remember, they're all crossed to that black point, the northern perennial. They all share a mother. And you can see that that mother phenotype actually is what the F1s show, which suggests that clonal growth is dominant. Okay, how about seed number or fecundity, fitness in the field? So now I have that all these same groups on the figure. I'm not going to walk through it stepwise. I've just got it all on here at once. And again, you can see F1s have this much higher phenotype. They're showing over-dominance for fecundity. And in this case, actually, our blue and green, our away phenotypes, tend to do the best. And this might be because in the southern part of the range, they're used to a drier climate where they've been selected for a higher seed set at the expense of that clonal growth. Okay, so let's actually put that together and see what's going on here with those traits. So now we've got clonal growth on our x-axis and seed number on our y-axis. And you can see that we do find that same pattern of the line here, but I've tricked you a little bit because our y-axis is not flowering time here, it's seed number. And so what we're actually seeing here is the opposite. We're seeing those F1s are benefiting from both sexual and asexual reproduction. So this suggests that there isn't that trade-off that we had previously identified, and somehow they're able to do both. Now, because we see this in the F1s, it's probably because we've recombined alleles that don't actually exist in nature. And so this suggests that you can actually do both, but selection over time doesn't select for that phenotype. So let's look at our F4s, all of those recombined individuals, not just F1s. Oh, sorry, I got one slide ahead of myself. I wanted to also quickly show you what's happening in year 2 here, so about overwintering. And you can see that the plants with more clonal growth have higher overwinter survival. So indeed, it looks like, as we predicted, that clonal strategy is about surviving into year 2. And then on the right-hand side of this figure, I'm showing you that the plants that had more clonal growth in year 1, so in their first year, produce more seeds in their second year. So if you make more vegetative rosettes in the first year, you have a higher chance of overwinter survival, and you have higher fecundity in the second year, which is exactly what we would predict from classic life history theory on perenniality and allocation trade-offs. Okay, so now let's go to those F4s. So these, remember, are a population where where we had 400 recombined individuals. And so here showing you a mean doesn't make much sense. And so what I've shown you instead is the entire distribution of phenotypes. And what you can really see is, as we would expect, they have a broad distribution of phenotypes because we're combining all these alleles from the different parents. In fact, the 3 sets of F4s don't differ much for clonal growth. And again, this suggests that clonal growth is dominant. And so even if you have the annual in red here as your one parent, provided your other parent is perennial, you're getting those clonal growth genes. But that's not the case for fecundity. So the annuals do tend to have lower fecundity. And then when we try and look across the 2 traits, again, we don't find that same trade-off. Plants are able to do both, suggesting that we can recombine alleles to have plants that both make a lot of seeds and make a lot of clones. We don't see those plants in nature, but we can artificially make them. Okay, so really we want to get at the genetic architecture of this, like I've mentioned to you. We want to try and understand how we now get these individuals that can have both of these strategies when we thought there was shared genetic architecture. So getting at the actual genes underlying these traits is going to really help us understand this pattern. As I said, unfortunately, I don't have that here for you today. But what we have data from, but it's not analyzed in pretty figures yet, is we've genotyped 300 individuals from all 3 sets of those F4 mapping populations. We're identifying regions of the genome that are involved in the sexual and clonal components of fitness to really try and get at the interactions between the genes, whether they're linked where there are single genes affecting multiple traits, which will allow us to characterize how these alleles are contributing to fitness and dominance relationships. Okay, so altogether, I hope that I've shown you that these annual and perennial ecotypes persist in near sympatry throughout their range with lots of recurrent gene flow, and yet there's strong divergence in phenotypes, which suggests that there's really strong selection and that we know that inversion is contributing to it. So genes map to that particular region of the genome. There's lots of variation maintained both within and between populations in terms of germination, flowering, and allocation. There's a mix of plastic environmental components to timing these things, and also genetic mechanisms that are maintained in different environments. And we're still trying to figure out this complex relationship between sexual and clonal growth. We thought we had a clear story of trade-offs between flowering and stolons. Our field data suggests that it's more complex, and it might have to do with the complexities of nature and being in the field, where depending on resources, other aspects of the environment, we're getting plants that are able to have sort of super fitness and do both of these strategies. So yeah, nature's complicated, and as we often see when we try and quantify fitness in the field, it gets a lot messier. So we're still trying to resolve those differences. All right, and with that, I would like to thank my current university, Queen's University in Canada, Syracuse University in the United States, where a lot of this work actually happened, and funding from NSERC currently and NSF formerly. And all of you for sticking with me today. Thank you.
Thanks, Janice. That was super interesting. I really enjoyed that. So thanks very much. I'm going to look at the chat to see if there are any questions just before I start asking. So I see Kenneth had a question. Kenneth, do you want to unmute yourself and ask, or should I read it out for you?
Hi, Kelsey, can you hear me?
Yep, can hear you fine.
Yeah, apologies, I don't have my microphone with me, so I hope the sound is good enough. I was just interested in the actual architecture of the plant, so the distribution of the meristems involved in clonal growth versus sexual reproduction. So are the same sets of meristems involved? So are the same meristems basically determined to become stolons or flowers, or are different meristems and different parts of the plant involved with this particular strategy? So is the apical meristem involved in floral formation and sort of basal axillary meristems for stolons?
Yeah, that's exactly right. So some work done about a decade or more ago by Pam Diggle and some of her students actually looked at the meristems and did, detailed morphology, which I would love to do, but don't do myself. And that's exactly right. So it looks like that the basal meristems have an indeterminate fate until flowering and bolting. And so they can either form stolons, or if the apical meristem transitions to flowering, and they have not made stolons yet, then they instead make flowering branches. So it still seems like the timing of transitioning to flowering is really important. And as I said, although I haven't myself looked at meristems under a microscope, I would love to. I just don't have the skills or someone in my lab to do that right now. We certainly see that borne out in the morphology. So, and in the plant architecture. So when you, the perennial plants have compressed internodes, and from the base of the plant, they form stolons from the loaf that come from meristems at the leaf axils, and they form stolons there and are, as I said, a sort of compressed rosette. The annuals will rapidly bolt, those same nodes will elongate, and then they'll eventually, if they live long enough, form flowering branches from those same meristems in the leaf axils. So it seems that they either make stolons or flowering, depending on the timing of bolting and presumably auxin and hormonal responses.
Thank you. That's really cool.
Yeah, I'd love to have someone in my lab who actually, knows how to do good microscopy and quantify these things, and then also get at some of the hormonal differences. So actually be able to quantify, whether this is an auxin response. We know gibberellic acid is important, and some of the genes we've identified with the QTL mapping approach point to some gibberellic acid response genes as well. So we know that that's probably involved too in those decisions.
Thank you very much.
Are there any other questions from folks? Oh, okay. Sorry.
That's okay.
It's part of the working from home thing. I actually do have a question. Oh, Glynis, I see your hand, so go ahead.
Thanks, Jenny. That was fantastic, really interesting. Amazing to have so many of your field plants out there and then be able to analyze them. That's really impressive. Wow, thanks. I'm sorry I lost connection for a short time, but I'll go back to the recording and listen to it fully. But I was wondering, have you thought of taking the F4 plants and planting them out back into some of the natural habitats and seeing how they then, how quickly they then become adapted to that sort of that seasonality, that moisture level?
Yeah, that would be great. It would be really wonderful to kind of see how strong, instead of a single common garden, to go back to their different environments and see how strong selection is there. It's just logistically challenging, right? So like in this slide that I have up with the photograph, that's what a natural population looks like. And so once they start growing clonally, it's really hard to tell who's who. And so when we did the field site in British Columbia, if you saw the photograph of the site, we actually cleared the site, put down black cloth to get rid of all the mimulus and other plants that were there, so we could very clearly quantify things. Obviously, that also is not ideal, but for practical reasons was kind of the only way we could do it. So I would love to do what you're saying. What we're doing right now, I have a student in my lab who's doing an artificial selection experiment in the greenhouse on clonal growth. So from a single population, she's selecting on low stolons and high stolons. And then, we've got a bunch of replicates and controls. She's currently on generation 3. And the goal of that is to see if we select on these different traits in artificial selection, what other traits, are we carrying along as a correlated response? And although her project will probably wrap up with the greenhouse plants, I'm hoping that a future student will take those selection lines and put that back in its home environment, because it's just one site. And then we can look at these plants that we artificially selected and see what happens to them. So somewhat related, but a little bit cleaner design, because it'll just be one site and we did selection instead of recombined. Part of the problem with the recombinant approach is every individual's genetically unique. So you do need these massive sample sizes for replication. Yeah.
Okay, great. Thank you. Thanks.
Sorry, that was a long answer.
I know, it was interesting. Thank you.
Denise, I have a question about the plants that you have that are making seeds and clones. And so I'm trying to remember what I've learned about mimulus and its outcrossing and selfing. Is there any sort of like divergence in the selfers and the outcrossers with those?
Yeah, so the species Mimulus guttatus, this species is a traditional mixed mater. So the outcrossing rate is about 0.65 on average, but it's really variable. It's self-compatible. It does suffer from some inbreeding depression, but not a ton. And the selfing probably happens partially from pollinators just being messy, but also delayed selfing. So lots of previous work from other folks in the 80s and 90s showed that if plants did not receive pollinator visits, then as the flower wilted, the anthers dragged over the stigma and deposited self-pollen. The annuals and perennials, I'm kind of smiling here because this is a question I get asked all the time, and I've been working on this species for over a decade now, and I don't know why I haven't just done this. We don't really know if the annuals and perennials differ in selfing and outcrossing. So some, again, older work by Kermit Ritland had a couple of annuals and a couple of perennials and tended to show the perennials were more highly outcrossing. They have bigger flowers and a larger anther stigma separation. So they have the morphology to suggest that they're going to be more outcrossing. But, which might be where you're going with the clonal growth, if they form these large clones, although they may not be autonomously selfing, they're going to be gynogamously selfing, right? So pollinators are going to be taking pollen from the clonal ramets back to each other. And so I actually suspect that the perennials are more highly selfing because of gytonogamy, but I don't have any evidence for that. So I really need to just do that project. I just haven't. But we actually don't know if there's real differences in the selfing rate between the annuals and perennials.
Okay, I would love to know. So hopefully we get a chance to do that project.
You know, if there's anyone older in the audience will remember back to the age of allozymes, which is all I need to get at that. I've got all the seed. If I could just do allozymes and a journal would still publish allozyme work, I could very easily get a selfing rate. But I don't have an allozyme lab, and so I have to use genetic markers, and it's, you know, actually way more time consuming and thousands of dollars more expensive. So we haven't done it.
Yeah. Totally fair. And then just related to the seeds with the fall and the spring cohorts, did that fall cohort, did they make it through the winter?
Yeah, so they overwinter perfectly well under snow.
Wow.
So yeah, so like I showed you in the middle part of the talk, those differences in germination and then those subsequent effects. So for example, that population, which is on Vancouver Island, we think it has this polymorphism. So they germinate in September, put on a few leaves, or August, like when the rains start, put on a few leaves and then just shut down under the snow through winter and then pick back up. At our actual field site where we planted fall and then spring, we actually lost our fall plants because the entire site flooded, which it does, like these are really wet environments, but all of our tiny little seeds or seedlings and tags basically floated to the edge of the field. And so they survived just fine, but we had just this like band of Mimulus plants and tags and couldn't track anyone. So we actually didn't get that data in the field from the overwintering because we lost our tags and plants, but they did okay, but they just moved.
Yeah, that's unfortunate. Oh, shame. Are there any other questions for Janice?
Don't think so.
I was wondering if you counted the seeds from the fall and spring cohorts, because you had flower number, but did you do seeds too?
Yeah, you know, because that experiment was this is one of the problems with greenhouse or growth chamber experiments. Seed number depends on pollinator visits. I mean, like I've said, they will set some self-seed, but I'm not sure it's really a good measure of fitness in a greenhouse. And so unfortunately, I mean, flower number is not a great measure of fitness either, because obviously you need to actually set seed, but I just feel like, yeah, it's too, it would have told us something different, which would have been the potential for self-fertilization.
Right.
Which might have been interesting, but yeah, no, we didn't do that.
Okay. All right. Any last questions? Or- All right. Thanks very much, Janice, for, oh, I saw a new message. Okay. Yeah, thanks very much for sharing your work with us. I really enjoyed listening to it, and it's great to chat to you and see you again, even if it's over Zoom. So thanks very much for doing this. Much appreciated.
Yeah, absolutely. Thank you. It was a pleasure, and thank you all for attending. It was a lot of fun.
Great. Thanks, everyone.
All right.